Topic, that is vectors. I know you must be thinking it's again this topic is very related to molecular cloning, and you will get the reason of going for this topic is later we can understand cloning in a much easier manner. That's why I have gone for this topic first, that is vectors. Here also in my earlier videos, when you are going for any mathematical calculation, anything, what you do is you need to learn the earlier steps. Without knowing those earlier steps, your next step can never be understood. So I always insist that in my earlier videos also you might feel that why man is saying so many times you could learn this, then you come to this, because the next lesson becomes very easy for you. So this is another lesson on recombinant DNA technology, and we are going for the lesson on vectors. The reason of choosing vectors is that I want to teach you cloning. Before I go for cloning, I must give you first knowledge will be based on restriction enzymes, endonuclease, and I hope you have watched my videos on restriction enzyme, endonuclease. More videos are coming up where we can talk about the details of endonuclease, the properties. So that details can be learned later. With that knowledge, we have come to this level. Now the vectors. What is a vector? A vector is a carrier or a delivering system, you can say. If I want to deliver something from somewhere, I need somebody who can do that work for me. So suppose, just for example, I want to deliver this pen and I want to give it to someone. I can do it myself, but if I want someone else to do it for me, then that is the work of the vector. So vectors are carriers. What they are carrying? The gene of interest. In this case, we are talking about recombinant DNA technology. Naturally, our interesting gene, that means the target gene or the gene of interest, where it was there, we have located, say for instance, this, I will take another color because we are going to write with blue. And first you try to understand what we are exactly up to, because details can be learned later, but the first basics should be learned. Now here is a particular chromosome and here are the nucleotide sequence. Now what I need to do is, I want to cut this point, cleavage, as I have said earlier that restriction enzyme, restriction digestion will take place where it will be cleaving, and this part will be cleaved and attached to some other part. So that delivering system is a vector. So in this case, who is going to deliver? It's a vector which is helping us in this job. And as I have said, a plasmid, a bacterial plasmid will help us in doing this job. And this is our vector, which is circular, double-stranded, and this is very commonly known vector is plasmid. So that cleavage cannot be at any point. It has to be a particular point. A particular system should be there where a vector is going to carry, and it will be introduced in some part in this vector, and where it will be attached, integrated, that part is known as the recombinant part, recombinant plasmid. Plasmid I have drawn circular, double-stranded, and this is going to act as a vector for us. Why? Because this is going to carry that target gene and it will be delivered to a cell, say a bacterial cell. The bacterial cell, I forgot to make it elliptical. This is the bacterial cell, and here I will be introducing this vector will carry my targeted gene, and from somewhere else this gene of interest was over here. It has to be cleaved at a particular point, say over here. The cleavage has to take place by the restriction enzyme endonuclease. It will be cleaving, and after cleaving it will be attached to this vector, which will be a carrier for me to carry the gene of interest into this bacterial cell. This is the bacterial cell where I will be introducing the vector In this case, as I have said, bacterial plasmids, which are common vectors for us carrying the targeted gene. So it will, that gene where our target gene will reach to some region, say here. This is our recombinant plasmid. This is the region where the gene of interest has been introduced into the vector, and this is going to be our recombinant plasmid. So this is the basic idea. What is behind all this mechanism and why I am learning today vectors. Because straight away I cannot enter the topic. I must have a particular question in my mind, and that question has to be understood, that why I am studying. We need a particular system. Why we need a particular system? Because it might happen that we are having this recombinant, we have to cut this and we have to cut this also. Because if we do not cleave this, we cannot introduce our gene of interest into it. We want someone, that is a vector, to carry the target gene for us, and that carrier, that is our vector. In this case, this is our vector which will carry, and as it is carrying this vector, say I have written the name of an expression vector at this stage, PBR322. It is there in your syllabus. If you check out NCERT, you will find this is there. You can take any other vector also. That's not the point. The point is that if I have taken an expression vector or whatever is the vector, the basic system is that target gene has to be cleaved. And here also it has to be cleaved, because without cleaving, how can I enter? Now, suppose I am cleaving here. Suppose I am cleaving here. Suppose I am cleaving here. So what it is happening? Finally, I will have so many cleavage points, and it will be losing. It will be under degradation. So that should not happen. So that means here also a particular point should be there. The cleavage point should not be many. The cleavage point should be one, where I will introduce my target gene, that is gene of interest. And where it will be introduced, there also a restriction enzyme has to be used. A restriction enzyme will cleave here. A restriction enzyme will cleave here also. And this is also a point I will put stars to make you understand that the same restriction enzyme endonuclease should be used. There are so many restriction enzymes there, but you need to use the same restriction enzyme endonuclease to cleave my target gene and to be introduced into the vector, so that I can prepare a recombinant plasmid. So the first point is same restriction enzyme. Now the next point, why I am using the same restriction enzyme? So that the compatibility is a factor. If it is not compatible, like you are using two phones and one is able to take up the Windows system and other is not able to take up, then it will not be compatible. Your transference, your files cannot be transferred properly. I hope you are understanding the point why I am saying it has to be the same restriction enzyme endonuclease which is cleaving my targeted gene, and here it is entering into the vector, and that also with the same restriction enzyme it has to be cleaved, and then it has to be ligated also. DNA ligase will help in, after cleaving it has to be attached. So DNA ligase, the mechanism should be there to attach. We have got our target gene and we have introduced it by cleavage, and then DNA ligase has ligated it. Now the vector is, you can say, the region where it is joining the vector, our target gene, where it is integrated into the vector, that place is known as recombinant plasmid. Now the next property, one is that restriction enzyme has cleaved. Second, very important property is it must have origin of replication. I must have my target gene introduced into the vector, and it should be able to make copies. So that if it is able to make copies, replication, that is copies. If it is making copies, then I can introduce those copies into my bacterial cell, where it is a host cell, you can say. This is my host cell. I have to introduce it into the host cell, and the host cell should be able to make multiple number of copies and carry on the task, where that is expression of the gene has to take place. That is the vectors, where it is talking about the expression vectors, we can talk about it. First is origin of replication. Let's say The vector, first it has to cleave, it has to take in the target gene, that is the recombinant it has formed, but it must have, each vector must have a region which is known as origin of replication. Without this, it cannot be called a vector only. Origin of replication should be there, where the protein binding will take place. Here in this place, you can see ROP, protein of replication. It will bind. Here, a protein will be binding to this origin of replication. Protein will be binding to this origin of replication. I will take another color. That means the protein will bind here. Protein binding, and DNA polymerase will be attaching itself. So this is the total mechanism I have. So I need origin of replication. Without origin of replication, it cannot be termed as a vector only. It cannot be that delivering system. About vectors, we have spoken about recombinant plasmid. Our gene of interest have been integrated into this region, and I can use a separate color to denote that yes, this is our recombinant region. Here is the origin of replication, without which we cannot call it a vector only. That is, the protein will be binding at this place, protein binding site, which is attached to this origin of replication. Then only the DNA polymerase will be attaching itself, and it recognizes. This is the basic idea. Next is that the particular spot where it is cleaving, I have said that I will use a compatible same restriction enzyme, that is endonuclease restriction. There are so many restriction enzyme endonucleases. We have to use a particular one or many, and it is at this spot. It is at this spot, you can say, the cloning site. That means the site where our gene of interest have been attached or integrated into the vector, and this site is a very important site. I will rub these parts because I need to use the place for other writing purposes. So it is at this spot. Let me rub the unnecessary part at this stage, because for explanation I need this part. Suppose at this place my gene of interest has been attached, that means cleavage is there. There are restriction enzymes, various. You can use the benefit of using 2 or 3 restriction enzyme endonuclease is that if EcoR1 is unable to cleave, EcoR1 has its own restriction signal sequence, and it will denote that sequence and it will cleave, and if it is unable to do that, then another one can do it, BAM, H1, or maybe HIN, D3. So this is the place where I found my multiple cloning site. I have said the cloning site. Cloning site is that site where the target gene has been introduced into the vector. It is the cloning site. So if I have many restriction enzymes, so it's a multiple cloning site. The benefit is that if one restriction enzyme is not able to find its own nucleotide signal sequence, then another one can find. If EcoR1 is not doing, then BAM H1 will do, otherwise HIN D3 will do. So this is the other factor. Now comes another very important factor, that is, first was origin of replication, where protein binding will take place, DNA polymerase attaches to it and recognizes. This is the fact over here. Second point, another point was about the restriction enzyme compatibility and then multiple cloning site. Now here I am talking about the properties of vectors. What is the other property? Can you guess what is the other property which is missing over here in this diagram? I think you have got it. If you are recollecting your memory, try to understand. There is a place where you will find the selectable marker is used. Here is the place where a selectable marker will be used with either antibiotic resistance gene or enzyme producing gene. Now why I need that selectable marker region? There is this is a question that we need to understand. Why we need a selectable marker? Of this selectable marker region is, suppose I say that I have a region where there is a mixture of my target gene. I have cleaved, remember I have cleaved my vector also, where that is the plasmid. Here in this case I am talking about the bacterial plasmids. So the cells are there which have been cleaved already, and I have cleaved the target gene also. So let me just take you to that place first, which I will explain, and then I will come to the selectable marker why I need it. Okay. So here there is a mixture and there is a buffer as usual present, and there are some target genes. I will take another color so that you can understand properly. These are the target genes which have been cleaved with the use of that restriction enzyme endonuclease, and these are the plasmids which have been cleaved. So in this case, let me use this color. I have 3 colors option, so I will use a red color, these cells. Now the question is, these are also cleaved, that is also cleaved, and when I am placing it in such a solution where there is DNA ligase, treating with ligase will ligate it, attach it. Now what happens is, there are 3 possibilities, and what might happen is that a part cuts from here, that means our target gene, a part cuts from there and attaches itself to these cells which have been ligated, and these are recombinant plasmids which I really want. So this is a possibility. Another possibility is also there, that it might happen there are target genes which have been cleaved. They will self-attach, means one is attaching with the other and they are self-annealing, joining. This is also a greater possibility, and that means self-ligated. And another possibility is there, that these cells which are left ligated, these will also join along with one another. That means they are becoming self-ligated. These plasmids which have been which have been cleaved, they will self-ligate. Like the target gene self-ligates. Here also, this is a recombinant case, which I really want. These only I have to introduce. So this into the host cell. So the question is that this may happen and this may happen. So I need a selectable marker which will carry the antibiotic resistance gene. There are 2 things that might happen. One is, either it will carry an antibiotic resistance gene, that you can take ampicillin resistance gene or tetracycline resistance gene, or it might also happen that it will carry enzyme producing gene. In that case, it is LAC-Z, that is encoding beta galactosidase. These are the 2 possibilities which is there in case of selectable marker region, so that I can select which cell has to be attached. Which cell? It's not all the cells. It has to be specifically some cells. Marker will help me to understand which cell I have to introduce into this vector. That is, in this case, very common vector is bacterial plasmid, smaller in size also. So here of course this is an expression vector I am showing, not only a cloning vector. So plasmids are cloning vectors. Let me rub this, otherwise you may ask me, ma'am, you have written this name, is this a cloning vector? No, that one was an expression vector. That also is a discussion, a point of discussion, a cloning vector and a expression vector. Right now I am on to this cloning vector only, and I am talking about the molecular cloning. So the basic thing, requirement, first requirement, origin of replication, so that it is able to make copies. Without that, it is no purpose of having a vector. Second requirement was that we must have multiple cloning site. A cloning site is the site where my target gene will be integrated into the vector. And if I have a multiple choice in this case, I can go for any restriction enzyme endonuclease. So that's also a point. The third point is a selectable marker region. Selectable marker region is a large stretch of nucleotide sequence which is present in the vector, and this region is carrying either, I am saying either antibiotic resistance gene, that is, let me write down so that you can note it down in your copies, that it is antibiotic resistance gene, and otherwise enzyme producing gene. So two things. If it is enzyme producing, then I am talking about LAC-Z. In that case, it will it will encode beta galactosidase. You can have a quick revision of that lactose lacoperon so that you can understand LAC-Z. Remember, that was beta galactosidase. So in this case, if it is enzyme producing, then LAC-Z. Otherwise, ampicillin or tetracycline, any antibiotic resistance gene. Now, what will happen if suppose I am introducing a particular target gene which carries already an ampicillin resistance gene in it, and it is introduced into this region. Now the question is, what will happen if it is introduced? If it is already having, if this vector is already having ampicillin resistance gene in it, then it will cause a disjuncture, you can say disrupt. It will disrupt. In this case, if it is not having, then it will be good enough. Suppose I am talking about these plasmids which have been self-ligated, and they are brought into a plate of bacterial cells where there are lots of ampicillin resistance gene present in it, then these will be growing. But in case of recombinant, where it is already having that ampicillin resistance gene, it will disrupt. So there is the point why we need selectable marker region, what is a selectable marker region, and why do we need it.